Okay, now we for now we have all this uh, female student. Okay, as usual. Okay, so feel free, Ken, when you are ready. Okay, so I just make a brief, quick introduction, and then it roll up.
Okay. So my name's Kenny Pence, excuse me, and let's see. I've been teaching biology for about 7 years now. Started back in 2016, right out of my grad school. Let's see. When I was in grad school, I actually focused on my topics were genetics was one of them. I focused on ecology and I focused on evolution. So those three things got me through my grad school uh, top or what do you call it, thesis paper. When I've been at Columbia State, I've kind of taught more human anatomy and physiology for the last 6 years, and then a little bit on the general bios, which would be your genetics and your cell biology and your microbiology. And I'm kind of excited to see um, how I do with the genetics for middle schoolers or middle school age students. So you want to go around and introduce yourselves?
They're usually shy, shy. Maybe what we can say, we can explain first why is genetic. It's very important. Okay, and then we can relate it with CRISPR. Okay, CRISPR,
CRISPR?
CRISPR,
Okay.
CRISPR. Yes, yes.
So genetics is the study of genes, and genes are little segments of DNA that make proteins for us. These proteins are going to shape our bodies. They're going to do functions for our bodies. So it's important to understand the segments of DNA for the genes. And if you understand the segments of DNA for those genes, we actually can manipulate those DNA segments into we can change them to do things that we want to. So, for example, normal people that don't have any diseases, they have what we call normal genes, and then there are some malfunctioning genes or defective genes out there that causes bad diseases in people. So, for example, have you ever heard of hemophilia or sickle cell anemia? Those are genes that aren't working correctly in the individual, causing them to have that disease. So, hemophilia is going to be uh, somebody will bleed more than they should. And sickle cell anemia is where their blood cells aren't circular, they're actually kind of moon shaped. And those are both caused by changes in DNA causing coming from the gene. And then with CRISPR is actually pretty new. It's a way that we can go in and locate these genes and change them to make them either work correctly, or sometimes we can add even more mistakes, and adding even more mistakes is going to turn them off. And then if we turn them off, the individual will no longer have that trait. Does that make sense? Never hear me?
Yes, we can.
Okay. And if you have anybody has any questions during this, just feel free to interrupt me and ask your question. So CRISPR, we actually discovered it from bacteria. The bacteria use CRISPR as a defense mechanism to chop up viral DNA. So viruses infect cells by inserting their DNA into the cell. So bacteria can take these CRISPR molecules and chop up viral DNA in order to protect themselves. So is everybody here wanting to go into the STEM or scientific field? Let me go ahead and share my screen for, let's see, the book information that we already have. Can everybody see my screen?
Yes.
Okay. Uh, so yeah, genetics is really important because again, genetics is the study of genes. Genes are DNA segments. So this is what DNA looks like. It's technically referred to as a double-stranded helix because it's two strands. So there's one over here and there's one over here, and it spirals around itself, making it that kind of twisted ladder case or ladder. And then these little lines you see in it, those are called nucleotides. We'll get to this later. But the arrangement of these colored strings is going to make a certain pattern, and that certain pattern is a gene. And that gene will code for a specific protein in our body. And again, the proteins will shape our bodies, and um, sometimes they do functions for our bodies, like again, making blood or making the enzymes in blood. So genetics is relatively a new science. It started probably in the 1960s, maybe a little bit early in the 1950s. But the science itself is um, stated or said to be created by Gregor Mendel. So Mendel is the father of genetics because of his famous experiments that he did back in the 1860s. What he did was extremely simple, and he actually got pretty lucky with his experiments. Uh, but a little bit on Gregor, he was a monk. Let's see, he actually went to school to become a math teacher. So he was very good at mathematics, and science was not his background. He never actually passed his test to become a teacher. So he went off and he lived at a monastery in Austria. And then at this monastery, he helped garden. He helped produce food. And then at the time, there was a lot of science going on, and most of the individuals that were doing research at the time were using rats as their go-to specimen. And Gregor, he tried a couple experiments with rats, but he didn't like it. He didn't like using rats. So he decided to use the plants in his garden for his experiments. It's very simple what he did. So he used pea plants, and he used pea plants because of so many traits that the plant actually has. It varies in the flower color from white to let's see, yellow. It varies in the seed colors from green to yellow. Like this individual he's using right now, this is what he calls a tall plant. And there are also some individuals that would barely get knee high, and he would consider those short individuals. Many other traits that he used. And again, he got kind of lucky because these traits are what we call simple Mendelian genetics because there's only one gene that's affecting these traits. But he actually went around and he hand fertilized or hand pollinated these flowers. So this part here, this is actually the female part of the flower. And then these parts here are actually the male parts of the flower. So he took off the pollen producing structures, which are the male parts, and then he went around and he hand chose what plants he wanted to fertilize together. That way he manipulated what the seeds or the offspring would be. Let's see, his experiments that he did, they didn't take very well at the time. So during this time, they knew that traits would pass on from parent to offspring, but they didn't know how exactly. So at the time, they called it the particle theory. There was some hidden particle inside of organisms that would pass on to the offspring, and those particles would change and manipulate the structure into a new organism. Again, nobody knew what they were at the time. So Mendel actually found out what they were, but with his experiments, nobody took him seriously, and he actually died well before his experiments became famous. So his experiments were brought back up in the 1950s and 1960s, and at the time we used his experiments to we redid everything he did, and we found out that the particle that we called at the time was a gene, and the gene is located in DNA. So these are some figures I have from my free textbook to use. Again, this is another image of Gregor Mendel, his real image. And then these are really good examples of his pea plants. So this individual has like a violet to white flower. Again, all he had to do was take off the female part, which is right there, and then the male parts are these little stigmas that are coming out with the pollen producing parts. He would cut those off, and then he would go around and hand pollinate all of the flowers. That way he could physically choose the parents of the seeds. And again, he was at the time he wasn't known or famous for his experiments. He became famous well after his death. About almost 100 years after his death was his experiments really relevant for us. So his famous experiment actually took a while. We have what's called an F1 and an F2. So F1, get my thing to work, equals parent generation. Okay, this is not working. Hang on. Let me do this. Parent grandchild. Okay, so his first round he did would be the F1 generation, which would be the normal parent generation. So this is where he went around and he took individuals that had yellow seeds and he had green seeds. So they started out with yellow seeds and green seeds. He let the individuals grow, and then he actually um, pollinated the two together. So the yellow pollen went to a green female part, and then the green pollen went to a yellow female part. He let the peas produce the seeds, and then he found that every seed that was produced from this F1 generation came out with the yellow seed color. So that didn't really tell him anything. He he noticed that all the individuals were given the yellow seed color. So with the F2 or the grandchild generation, all of those yellow seeds that he acquired, he allowed them to, he dried them out a little bit, and then he planted these yellow seeds. And what he found out is the individuals that were produced by the yellow seeds, they came up with some of them being yellow seeds themselves, or some of them producing the green seeds. So he found out that the green seeds from the parent generation came back in the grandchild generation. So that was showing him that there was something hidden inside of the yellow seeds that allowed for the green seeds to reoccur. So again, that's that little particle that they were trying to find. And then over here, this is showing you the particle and how it's explained. So inside of the individual plant, there are two particles. Every one of the plants has two particles, either a yellow or a green. So the yellow one somehow left the yellow individual plants and then went over into either the pollen or the seed, and then when it was combined together with the other pollen or the seed, whichever one it was used to fertilize with, the individual gathered up the yellow particle. And then after the F2 generation, they found that the green particle came back. So that means that Gregor determines that there's always two particles. We'll get to this a little bit later, but one of them is called a dominant gene, and the other one's called a recessive gene. So if the individual gets a dominant gene, the dominant gene always overshadows or overpowers the recessive gene. In this case, you see the Y is a big Y. That means that big Y is a recessive gene. I mean, recessive, my bad. It's a dominant gene. And the lowercase g over here is a recessive gene. That means this individual, that since it got a Y, it's going to be yellow. And since it didn't get the Y, it's going to, let's see. Yeah, they're using two different letters. So this individual that got a Y is going to be yellow. The only way to get a green is to get two G's. So if individual gets a Y in any of these combinations, that individual is always going to be yellow. So with this one here, YG, you got the Y, it's yellow. All these were Y, so these would be yellow. And then with the two small lowercase g's, all these individuals will be green. We actually have some traits like this in us. So one would be detached and attached earlobes. So if your earlobes are attached and they don't hang, that is a recessive trait. But if your earlobes hang a little bit and they don't attach directly to your skin, that's a recessive trait. Do you have any questions?
So the parent is coming from the original plant. Is that correct?
Yes.
Uh-huh.
Let me see.
So basically, when we have this plant we see around us, is that called the parents? All of them parents?
So let's see. It's kind of hard to see from the image, but so everything above this line would be the parent. So these seeds were planted, and then these seeds produce these flowers. So that would be the parent generation. Okay. And then these individuals produce seeds, these seeds here, right? Okay, so these seeds here would technically be like the grandchild of the parent, because these seeds are coming from the individuals made by this this growing. That makes sense?
Okay, so the grandchild coming from the first seed, right? The first that we plant.
So okay, the grandchilds are okay. Let's see. These are the parent. So these are the parent seeds, correct?
Right.
Okay. So these technically are the child seeds. Okay.
Right. Uh-huh.
And then these are the grandchild seeds. That makes sense? Yeah. So you get the parent seeds up here that are being planted the first time. These right here are the child seeds. And then these over here that are produced are the grandchild seeds. That makes sense?
Yeah. So that need to be come from the same spot, the same parent planted in a spot, right? In one spot.
Yes.
And then from that spot, you generate, or it's coming, or it yield grandchild, right?
Yeah.
From
yes.
the same, okay.
From the same line, yes. Same line.
From the same line. Now, if we have two location or two pot, right? So let's suppose that we plant it at, in a pot, or, a small pot, okay? If we have two pots, then we have two parents, correct?
Mm-hmm.
All right.
Yes.
And then from that two parent, then we have a lot of grandchild, many of them.
Yes. Thousands. Mm-hmm.
Hmm. Now, uh, if we relate it to human, how did works? How did work? Is that parent
Okay. Related to
uh,
human. I can do that.
exactly the same?
Well, again, Gregor got a little lucky because the traits that he was looking at, we call simple Mendelian genetics now. With humans and other organisms, it's a little bit more complicated than this. But
Mm,
yes, normally all of our genes that we have in us came from our parents, and our
yes.
parents' genes came from our grandparents.
Right, right, right,
Yes.
right.
So
Now, yeah, go ahead.
So let's say that's I can kind of simplify it, but doesn't again, it's not really how it works because hair color is a little bit more than simplistic. But let's say somebody had black hair and somebody has blonde hair.
Right,
So the individual that was, let's say that your grandparents had one had black and one had blonde, and then your parent that came out from the your grandparents was a black haired individual.
right, right.
Okay, and then your parents both have black hair, but when you
Mm,
were born, you came out with blonde hair. That's
mm.
because the blonde hair gene was still inside of DNA of one of your parents.
Okay, so that called the strongest gene, correct? The strongest gene.
The
I think
strongest gene wins. Yes, dominant.
you the dominant. Okay,
Dominant gene, yes.
right, right, right. Now, this is very important. During the process, how many of the gene will will be disappear? The weakest one. Okay, say from one parent, from the parent to go to grandchild. Is there any, can we predict how many percent of the gene will be gone?
Yes, we can do that based on a Punnett square. You mean do that today?
No, no, this is great. I mean, I just kind of,
Yeah,
give
we can
this
predict that. Yes, we can do
yes,
something called a Punnett square, and we can use a
predict.
Punnett square to predict the chances of getting a gene. Yes.
And this is very important. Okay, I think this is very important. For example, okay, now this is springtime. Okay, springtime always great if we look at the flowers. However, the negative effect will be if somebody, you know, have problem with the pollen. Okay, so we call it. It will be sneezing all the time, sneeze or coughing and all of that. Now, if my parent, okay, have that, unfortunate allergies to a certain tree, say oak tree, say like, a pears, okay, pear trees. All right. Now, I think most of you already seen the pear trees is the one that look like white on the YouTube channel that Harry, okay, share it to you. So this is important, kid. Anything that we share, okay, in that YouTube channel, all right, has a meaning, has a meaning. Let's suppose, okay, the my parent has a problem, will be sneezing with the pollen that you know come from the pear trees. All right, the white trees that I show on the YouTube channel. What will be the probability? Okay, that I will inherit the same disease. Yeah, we call it disease or unfortunate situation. Okay. Now, Professor Ken already say we can predict. It is incredible. Okay. What predict mean? We can tell. Okay. In the next generation, maybe my grandson, okay, or the great great grandson from my parent will inherit the same problem with the allergy, okay, from the pear trees or peanut butter or whatever it is. All right. Now, This gene is very significant. So you guys are lucky. Okay, Professor Ken explain all of that. Look, even in my situation, okay, I'm not in the area of STEM. I'm economic. Okay, but the recent development in this gene, all right, treatment is so significant. It will change the global way of thought. Okay, how we treat diseases. Okay, so you guys, okay, 10 of them, okay, 13 participants, minus two of us, maybe minus Professor Ken and I. So 11 kids. It's very, very lucky to hear all of this. All right. So what is the application of it? The application is very important. Okay. You may start now to, okay, to research what is the problem if, let's say, for example, this will be unfortunate example, okay? Let's suppose your grandfather or great-great-great-grandfather, okay? The fourth generation, all right? So the first generation die because the heart disease, all right? Now the second one also disappear because of the heart disease, all right? I will not say about these two. Now, you are the sixth generation. You see? One, two, three, four, five, six. You are here. Let's suppose this. All right. Okay. Student A. Student A. Okay. So, grandparent from generation one and two die because the heart attack. Okay. So, what is the probability the student now still, the student like you, still in the elementary school, okay, sixth grade, will 50 years from now also die because of the heart disease? Can we predict that, Professor Ken?
If it's a disease that's caused by a gene, yes.
Yes. You see? I mean, it's just totally incredible. Why? Okay. Why such a great, okay, approach? Okay. Delay so many years until CRISPR come out. Why is that, Professor Ken? What do you think?
Well, again, CRISPR's pretty new, within the last 10 to 15 years. So we're still learning it ourselves. The thing with CRISPR is it's got two specific parts. It's got a locator and a cutter. So we have, for us to use it on ourselves or for our general benefit, we have to figure out where we want to cut and then how to cut it. So,
Mhm.
and there's hundreds of different types of CRISPRs too. So we have
Right.
to figure out the right one for the right circumstance.
Okay.
And we're still learning that right now. But if we can figure out, again, most of our disease genes, we should be able to combat those in our own cells.
Right. Now let's take an example in our example here. Okay. Gregor, Mendelian genetic. Now, if we know after this experiment, okay, we know which is the weakest one, right? You say that on the top is the strongest gene, dominant gene.
Dominant overpowers the recessive, yes.
Okay. So if I would like to generate the next one, okay, for the grandchild that really, really good, okay, can I take the weakest gene over there and replace and make all stronger gene or dominant gene? Is that possible?
Um, well, technically yes, but what you're saying is the dominant gene is the better of the genes. That's
Yes.
not necessarily true. Sometimes
Oh,
a dominant gene can be a bad gene.
ah,
Yeah. So I'm trying to think of one. Um, humans.
yeah, 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 yeah.
Not Huntington's.
The peanut butter allergy. Okay.
Oh, what's it called?
So allergy
Uh,
to peanut butter. Okay, go ahead.
well, muscular dystrophy. Muscular dystrophy. That's a dominant gene. So if somebody has a gene for muscular dystrophy, it will always be there. And we consider that a dominant because it always shows up. So
Okay.
the dominant doesn't necessarily mean it's the better of the genes.
The beta gene.
Yes, the
Okay.
recessive just means that it's hidden if a dominant shows up.
So it will be miserable if I inherit dominant gene but bad one.
But
Correct?
that's correct. Yes, it will always a dominant disease that causes a gene will always show up in the parent and the child. So this is one
Ah,
of those that shows up every generation if they get the dominant gene.
wow. Okay. So what I can see right now, all right, practically speaking, okay, but in reality, it will be difficult. So everyone on this planet Earth, if possible, need to be tested. What is the bad gene that they carry? Am I correct?
Yes, we do that. Yes, it's called screening. We do screens for individuals. A lot of our U.S. Fertility clinics,
Mm.
when two individuals want to have a baby, they would go
Oh.
get their genes screened to see if they have any sort of hidden recessive traits. Because if
Mm.
they had a dominant, they would know. But they get their DNA screened to see if they have any hidden recessive traits. And then once we
Mm.
screen them, we can do that Punnett square I talked about, and we
Yes.
can find the probability of the child getting one of those recessive traits.
Yeah. So, excuse me, I'm going kind of explain it in Indonesia, okay, in
Okay.
bahasa.
Okay.
Alright. Now, adik-adik ya, kalian sudah dengar ini dalam diskusi dalam bahasa Inggris, sehari juga belajar. Sehari juga belajar. Artinya, genetik yang kalian lagi belajar ini is very important. Kenapa? Karena ini akan mempengaruhi bagaimana seorang dokter di waktu yang akan datang in the future, a doctor, a medical doctor, or anybody that work on this area, okay, can help, bisa membantu orang sakit jantung. Jadi tadi sehari bilang, katakan ada 6 kalian, katakan 6 generasi, ya. Nah, maaf, sehari ambil aja generasi ke-6 tuh Hani. Hani, ya. Kamu generasi ke-6. Oke. Nah, generasi 1, 2, 3, 4, 5 ini kan engkong-engkong ya. Ya, betul nggak ya? Ini, eh, Ibu Lisnawati ya. Oke. Jadi engkong yang pertama, oke, pertama meninggal itu karena penyakit jantung. Oke, yang kedua meninggal karena penyakit jantung. Ya, jadi bagaimana Hani yang sekarang ini, ya, maaf ya Hani, kamu akan apa? Panjang umur. Ini hanya example, ya. Ini aja contoh, ya. Nah, bagaimana Hani yang generasi ke-6 ini, apakah dia akan meninggal juga karena sakit jantung, ya? Jadi 50 tahun sebelum kejadian ini, kita sudah bisa ya mengestimasi gitu loh. Apakah ini akan meninggal juga oleh sakit jantung? Kalau iya, makanya tadi Profesor Kenny bilang, we can estimate what is the probability that you will be die because of the gene, the bad gene carry out from your great grandparent. Okay, so this is very important. Yeah. Therefore, guys, you are lucky today. None of your school talk about this. Okay. Now you know in the future, di masa depan, ya, hal ini akan sangat penting. Oke. Bayangkan saja kalau kamu menjadi dokter yang bisa mempredik ini, semua orang akan datang ke kamu. Kamu seperti ahli nujum. Oke. Yang bisa tahu bilang, "Oh, kamu Mr. A atau Mr. B, okay, you will be die, okay, because of the disease." Okay. Nah, ini. So, this is very important. Okay. Now, in this video, Michelle Tang, okay, Kak Michelle. Michelle Tang will also work in this area. She has been admitted into Colorado. Okay, Michelle, can you introduce yourself? Okay. Hello, Michelle. Just say hello to everyone.
Yeah. Good evening, sir.
Okay. So, all right. So this is
Hello.
Michelle. Okay. So what will happen with Michelle? If she work in one of this area, she will be the best medical doctor or the best researcher who can find out what is going on. So the world, dunia ini, the world, okay, faces so many problems. Okay, let's take an example. Okay, Professor Ken maybe can let us know. Let's take an example about Parkinson. Ken, is there any cure for Parkinson? Medication for
Medication,
Parkinson?
yes, but no cure.
No
They can,
cure.
yeah, no cure.
So, do you think that CRISPR, the genetic approach, will be able to overcome this problem, Parkinson?
It might. Again, I'd have to see if Parkinson is caused by one specific gene. If it is, then maybe CRISPR could help. The thing is, with CRISPR in a human that's already formed, it's kind of hard to change DNA in a human that's already been created because our DNA in every
Oh,
cell is the same. So the thing with
oh,
you can do it. It's been shown that you can change some cells, but the thing is you can't change every cell because every
mm,
cell's got their own DNA.
right.
If
Yeah,
you
yeah,
were
yeah, yeah.
like if you can change the DNA from the sperm or the egg cell that fuse and make the individual, then that individual will have every cell in their body changed because of that one cell.
Oh,
But
okay.
but there are examples of CRISPR helping individuals that are formed already. It's just they
Yeah,
have to keep doing it over and over again.
yeah, yeah. So there will be a chain effect, right?
Hmm.
If you take out one, it will affect the other one. Okay. So this is a great, great area for research. This is a great area for future research. It's just totally incredible. Okay, a lot of people just want to do the same thing, but this is a new thing, revolutionize everything. Okay, so that's the reason why I can. The purpose of this, Professor Ken, the purpose is our discussion today is nothing but to get the new, okay, the new brain, the new kid, okay, to help the world, okay, to find all this possibility for the good of human being, okay, for the good of human being. All right, Professor Ken and me or anybody else just do not, it just not possible for us to come back to high school, okay, and to learn all of this. All right. Okay. Therefore, we need Michelle. Okay. We need Honey. We had Noel. Okay. Louis and everybody else. Now, my suggestion to you guys is this. All right. If you really want to make a difference to this world, okay, we have here 15 minus 2, 13 children. All right. Jika kamu ingin membuat sesuatu yang sangat baik, tidak saja buat kemanusiaan, but also for your career, this is it. This is it. Okay? You have to learn and study this area. And we are hoping Professor Ken will not just talk about genetic. We will make progress. We start talking about DNA. We just start talking about somebody else. In years to come, let's take an example. If you are in the 6th grade now, all right? Now, before high school, you have 6 years. And we are hoping that, okay, Professor Ken, all right, will help you in the next 6 years, but we are not sure because, okay, Professor Ken is busy. He got a full-time job, all right, and the most important, he volunteered his time, guys. 8 o'clock in the morning in the United States. All right. I used to, and it's Saturday, I used to wake up at 10 a.m., right? Professor Ken. We sacrifice our time for you guys. Okay. So let's come back. Okay. I can talk forever about this. Okay. Let Professor Ken continue. Okay, his lecture. Thank you.
Is there any questions that I can answer? But this is all that we should go over today, because what's the next one? Let me look at the next page.
I think
Yeah,
Michelle
the next
can.
page. The next page is it looks like it's cell gene to protein process, which is going to take it's a whole lot more in depth than this is.
Yes.
Let me see if I can come up with some more examples of recessive and dominant. Again, there's not that many in the human that's just simplistic Mendelian. A lot of, again, the reason why Gregor got lucky is because with these pea plants, these are monohybrid or monogenetic, meaning that one gene for one trait. There's also something called polygenetic. Polygenetic means there's multiple genes that are going to cause the trait. So that's what most of our genes are in our bodies, that we have multiple segments of DNA that change us and form the way we look. Like blue eye color is several genes. Skin color is actually several genes combined together. But you can, if we know these genes and the combinations of these genes, we can do what's called a Punnett square. And a Punnett square will show you the probability of you passing on a gene combined with the other parent. It's important for some individuals who are what are called carriers, individuals that know they have, like, let's say somebody's parent had a disease, but they didn't get the disease. There's a chance that they still have the gene in them for that disease. So if they were to have a child, there is a chance that their child could get that disease that their parent had because they still have the gene inside of them. So this is what your jobs would be as a geneticist, was to screen that individual to see if they have that gene, and if they do, you could give them a percentage of the chance that their child could wind up getting the same disease that their parents had.
Yeah, this is incredible. Jadi, okay, yang dikatakan oleh Profesor Ken, ya, gene ini bisa lompat. So it basically can mutate, right? For example, my parent has a problem. Okay, let's take an example that I have before. The first grandpa, the second grandpa died because of the heart disease. Okay, but the third and the fourth is fine. Okay, now Professor Ken said that is possible. However, the bad gene is still there. Okay, and the fifth one will inherit it too. Am I correct? Okay.
Correct. Yes.
Okay. All right. Now, the next thing, okay, how many generations? Okay. I use an example as honey. Okay. Okay, for sure, we can predict, okay, when it will coming. So does it also mean that about the generation, which generation, okay, will inherit, and they already had 6 generations in inherited, but the third and the fourth, all right, it did not pop up on them. It did not appear. But the fifth and the sixth, it might. So, okay, has there any study that can predict exactly if for peanut butter allergy, okay, it is always the third generation will have it. Okay, 1, 2, 1 has it, 2, 3, 4, it's free, and the fourth generation, okay, will inherit it. Jadi sehari tanya, ya, kalau orang yang alergi kacang itu generasi 1, ya, the allergy. Apakah kita bisa menyatakan kedua, ketiga, keempat itu free dan dia selalu muncul keempat, generasi keempat. I was, asking, will that is possible, okay, to predict exactly for a certain disease, which generation, okay, after how many generation it will pop up.
I'm
Okay.
looking to see if peanut allergies are dominant or recessive. Let's see.
If it is dominant, you cannot get rid of it, right?
Correct, yes. It would make sense if it's recessive.
So we will learn a lot, okay, during this process, okay? And we will learn a lot in this class. And now, we are fortunate we have Sister Florence with us. Okay, Sister Florence is a Catholic sister.
Hi,
Okay, silakan
I'm
Ibu.
from Flores.
Okay.
Okay,
Suster,
Okay.
silakan Suster ngomong dalam bahasa Indonesia nggak apa-apa, Suster, silakan.
it's okay. I'm Florencia from Flores. I live in Momere. I have been in Poland, but we didn't use English in Poland because we speak in Poland Polish language. So now I am here in Momere, but I would like to improve my English. So I joined this meeting. Yes, and then I just want to improve my English because English is now it's very important, necessary to learn more and more. Only that I will say now, and then thank you very much for sending me this link, and then I hope I can improve my English. By the way, now many greetings from students in the high school Bakpiarsa. They would like to learn English also, but not too many because they also have a lot of subject here. They have to learn mathematics and so on. Okay, many greetings. Thank you.
Yeah, thank you.
Thank you.
Thank you. Yeah. Okay, go ahead, Ken. You see this, sister explain. All right. So, all right. Sister Florence coming from, Eastern Indonesia with the, the facility is, is lower than on the west side, West Indonesia. Okay. So we are here to help everybody, to help them. Like Sister Florence said, the children would like to learn English, but they cannot learn other things because so many classes that they have. So what we will do, sister, okay, we will do whatever we can, all right? And what you need, okay, we need your help as well. Tell your children to come at every English classes that we offer, okay? So we are very, very happy to help anybody, okay? Especially from the places where it's so difficult to find someone, okay, who can teach English. This is about human resources, okay? See, all right, Professor Ken, you see that there's a lot of problems, there's a lot of challenges, but, okay, we will fix it one by one. Is there any special request, sister? What can we do for your school? Is there anything that we can do for your school?
Okay, thank you.
Ya, apakah ada sesuatu program yang khusus yang perlu kita sampaikan, berikan kepada murid-murid, Sister Florence, untuk belajar bahasa Inggris?
Okay, I will do it. I will try to help them.
Ya, oke. Ask them. It is all level. Suster punya sekolah itu dari SD sampai SMA, betul tidak?
TK.
Dari TK
ya,
sampai SMA
iya.
di Maumere. Berapa
Iya.
studentnya? Ada berapa semua? Kalau kita total ada 1.000 orang dari TK.
Di SMA, sorry, di SMA ada berapa orang? Kamu semua berapa orang SMA? Hah? Seluruh SMA. Bagian. Hah? Di SD hampir 300 atau 400-an.
Ya.
SMA 500. Saya belum terlalu tahu persis jumlahnya.
Yeah. So in total we have 800 children. Professor Ken.
Yes.
We have 800 students who would like to learn English. You see that? Ya, kita akan bantu 800 anak. Okay. Now, suster punya komputer nggak? Punya komputer? Do you have computer? They don't have computer. Okay.
I don't have.
Yeah. And you see, this is okay. This is real. The challenge. You see that, Professor Ken? The challenge is real. There's no computer. They cannot learn. Okay. So
I only have mobile phone. So
only one cell phone. Like the sister is using that now. Okay, sister. Nanti kita akan bicara ya. Setelah ini kita akan bicara. Oke. Aku akan nanti WA. Kita akan nanti bicara dengan suster. Habis ini 5 menit 10 menit lagi ya. Kita akan set up sesuatu yang khusus buat suster. Okay, sorry Ken for the interruption.
You're fine. You're fine.
It's okay. Never mind. Sir.
Yeah. So next week, what will be the next week? Next week we will talk about the third pages and the fourth pages. Is that correct?
4 and 5.
4 and 5.
Yeah, let's do 4 and 5.
Okay. Okay. 3, 4, and 5, and I will put the material in here. All right. I think Michelle. Okay. Michelle. Okay. Do you have any question or anybody else? All right. Do you have any question?
Let me stop sharing the screen, see everybody.
Oh, Michelle left.
Wow.
Okay. All right. Finka. All right. Finka, kamu ada pertanyaan nggak? Do you have any question? Okay. Do you understand what we are discussing today? Secara umum. Okay. How many of you understood what Professor Ken discussed? And we all discussed. Okay. You just raise your hand like this. Raise your hand. Okay. Okay, Luis, kamu ngerti? Okay, taruh di kamera. Okay, Michelle, you understand? Finka ngerti nggak? Finka, kasih tangan gini kalau ngerti. Kasih tangan. Okay, Noel. Okay. So most of them understood what you said. Most of them understood. Okay. All right, Michelle, you are in this area. Do you have any question? All right. General question. Go ahead, Michelle.
I think we have a question.
Okay. Oh. All right. Sister, kalau bisa di-mute dulu, Sister. Sister Florence di-mute dulu supaya yang lain bisa dengar. Okay, Michelle. When you talk louder on your question.
Okay, sir. My question is that for some reason there's genetic genes. I think my question revolves around the dominant and recessive. For example, my grandfather from my mother's side, let's just say that, has curly hair or wavy, I must say. And then my mother and then her other siblings all have straight hair, but somehow passed down to me, and now I have curly hair. And my additional information is that my father possesses straight hair, and I have really curly hair, while my grandfather is only have wavy hair. Is that sort of a dominant gene that passed down and skipped generations somehow?
So what you're talking about is a process called codominance. Codominance is, or well, it could be, it's either codominance or incomplete dominance. I'd have to get the information again, but with one of the two, there's either two dominant traits that are are both showing together, and they're kind of mixing. So you have really curly hair plus straight hair make wavy hair. Does that make sense? So if you get there's a curly hair gene and a straight hair gene, and if the two come together, that kind of combine for a new trait to make wavy hair. Does that make sense?
Oh, it does. But the confusing part is that the one that has wavy hair is actually my grandfather, and it somehow inherited to me, skip
Mhm.
generations, because my mom and her other siblings doesn't have any curly or wavy hair.
Okay.
And I have really curly, and yeah.
Okay, so
So
wavy.
the curly
Let me
hairs,
do something real quick. I'll do H and H. So it looks like it's incomplete dominant. So your grandfather has wavy hair, correct? And your mother has straight?
correct, and my father also straight hair.
Okay, so your mother is going to be HH. Okay, so it's straight. This is what I was talking about, where it's not simplistic Mendelian. So there's probably something else going on in there, that the trait that your grandfather had for his wavy hair was passed to your mother, and then your mother passed it to you, and then along with your father's gene, it made your hair curly, correct? Your curly hair, wavy hair,
Yeah.
curly hair.
Yes.
Yeah.
Yeah,
So
that's the case.
uh, wavy, curly. So yeah, it looks like your father has normal straight hair.
Yes, correct.
Yeah, it looks like you got a. Okay, let's do. I can do backwards. Let's say, okay, so straight hair is dominant. Yes, your mother's heterozygous. That makes sense. Your mother is heterozygous
Yes.
because she has your father's recessive, or your grandfather's recessive curly gene or wavy gene, and then you got.
Yes.
Looks like you're going to be recessive. Your father is also homozygous because you didn't get the
Yes.
dominant gene. Yes, this makes sense. So your parents are both heterozygous for the the trait, and you looks like you got both recessive genes for the wavy hair. But there is again, Mendelian genetics is not a lot of genes to look at in humans, but again, there's examples of Mendelian genetics in other organisms. With humans, it's a little bit more complicated in this simplistic Mendelian. There's not a lot of just dominant and recessive traits in humans. It's a whole combination of different factors.
Yeah, this is great, guys. Did you notice? Okay, when Michelle asked the question, and then okay, Professor Ken start writing. That how she predict. That how he predict. Okay, all right. That's the way how you say okay from dominant and gene. All right, you will learn that. Will they learn about that too?
Yes, later on in the book, I can see Punnett squares.
Oh, okay. It is exciting. Okay. Yeah, it is exciting. Now, you guys, okay, can tell for sure. You can start working on your family and see, and then you can start predict. And Professor Ken will tell you the formula to do that. Okay, I'm so excited. Okay, we are 2 minutes past already here. Okay, I don't want to take the most valuable time for Professor Ken. Is there any, is I pass it to you again, Ken?
If nobody's any questions, I will be here next week, and we'll look at what's called the dogma. We'll look how genes are created from DNA.
Okay.
Just nice to meet
Fantastic.
everybody.
Okay. Are there any questions from the participants? If not, okay, maybe we need to to end this meeting because Professor Ken has some other things, that going on, okay, this morning. So again, we are lucky. We appreciate. Okay, now before we leave, okay, can anybody tell a very simple gesture to Professor Ken? Okay, just say thank you to Professor Ken. Okay, open your, open your mic and say thank you. All right, and then we will close this. Okay, go ahead. Who want to do first?
Thank you.
That's it.
Thank you.
Come on,
You guys don't have to
don't
be
be
shy.
shy.
You don't have to be shy.
Yeah, Noel. Okay, say thank you. Finca, you have to practice English here, okay? Okay, Mariana, say something. All right, we don't want to wait too long here. Okay. Okay, they may be shy. They're afraid.
That's
Okay, their
okay.
English
That's
is not
fine. They
good
don't have
enough.
to. That's
Okay,
fine.
that's good enough. Okay, at least they can say bye. Okay.
Okay, I'll see you next week.
Okay, thank you, Ken.
Yeah.
Bye-bye.
Bye.
Okay, thank you.
Bye.
Thank
You're
you.
welcome.
Kita akan bicara ya, Suster. Okay.
Thank you, sir.